Okay, cool. We're recording. Yeah, I'll talk about our genomic surveillance of RSV that we've been doing for going on 3 years now. So I thought I'd just kind of broadly begin with an introduction to our infectious disease laboratory within our Department of Health. I know every Department of Health is kind of a unique thing, so I thought I'd just mention where we're positioned. So we have within our infectious disease lab a number of different units. Um, etc. These units then, are speaking out with different outside groups, right? So if anything, CDC to our department epidemiologists to tribal groups, etc. And we're positioned here within a dedicated sequencing and bioinformatics subgroup. Um, so here's us. It's a bit of an outdated picture, but it's our little unit. And I thought I'd specifically introduce our RSV core group. We're kind of adding people to this, but these are the people who've been doing the most work here. Um, Sarah, who's pictured in the front right, really got everything started. She's now our sequencing and bioinformatics supervisor, but got started a few years back before she assumed that role with the RSV sequencing project. And then I wanted to highlight myself and Dan Evans in the back there with the green circles. We've been kind of tag teaming this. Dan is a very skilled genomic epidemiologist and has been taking the data that we're generating and providing a lot of insights and kind of advancing the ball a lot. Just a brief bit of background. I'm sure many of you are familiar with respiratory syncytial virus, human respiratory syncytial virus specifically, but it's a virus that tends to cause more severe disease in young children, elderly, or immunocompromised people. In Minnesota, we tend to see about 3,000 hospitalizations each respiratory season. So it does put some amount of strain on our medical systems here. Um, the RSV virus is a single-stranded RNA genome. It's about 15,000 nucleotides long, almost exactly half the length of the COVID virus genome, and in many ways has a lot of similarities. It does behave differently once it's in a cell, an infected cell. It'll cause fusion with surrounding cells and form kind of this consolidated cell called a syncytium. That's where it gets its name. I'm not a virologist, so don't ask me too much more about the details. Um, and RSV tends to fall into two major subgroups, RSV A and B. They share roughly about 70% of their amino acids are common between the group, but they do have some key differences. Many of them fall within their um, G and F, the glycophilic and F protein that are responsible for a lot of the cell recognition and adhesion properties and tend to be the kind of highly mutagenic sites. A little bit on our genomic epi side. Not all laboratory confirmed RSV cases must be reported to MDH. Um, we are we do receive reports from all hospitalized cases, um, but we rarely get any subgroup data. We might get whether there was a PCR test for A or B, but really little detail beyond that. And we really can't perform much outbreak responses because we're already capturing, or we're only capturing the data once, um, we have a hospitalization. In some ways, we're not able to kind of predict or react quickly to potential outbreaks. Um, so what that leaves us is a situation where we really have to do a lot of the groundwork in terms of seeking out these genomes if we want to do any genomic analyses or be able to predict or respond to outbreaks a little bit faster. And so uh, at the beginning of this project, Dan and Sarah and I proposed this idea of can we collaborate across the lab epi interface to strengthen RSV surveillance by using whole genome sequencing technology. Something kind of a question that I think a lot of us are familiar with COVID sequencing. Um, but we thought we could expand it to RSV. So we thought we could perform more precise surveillance. We could maybe detect clusters and outbreaks We could identify new variants and lineages, and we could potentially start the process of associating specific mutations or specific subgroups with disease risk or vaccine escape or treatment resistance, that sort of thing. So, where we are now, I'm going to talk mostly about our first 2 years of sequencing efforts. We're now in the early stages of our third, really what will end up being a complete respiratory season here. So, some of these numbers might be a little out of date, but it still is going to, you'll see it's a good amount of data that we're going to walk through. Um, so we have received specimens or had at the end of the last respiratory season from around 29 healthcare facilities. This was a lot of work from Dan and from our epidemiologists just to kind of get the ball rolling, telling them we are interested in RSV that you see, please send us samples. We want to sequence them. So I think our first year we had maybe like 11 or 12 submitters, and then we really expanded that in the second year up to 29. I'm certain above that now. Um, in terms of how we're sequencing it, we're performing a tiled amplicon approach. It's a very commonly used approach for COVID, so you might be familiar with that. But essentially sequencing small chunks of the genome that partially overlap with each neighboring chunk across the entire genome. Each one has a dedicated set of primers that we amplify, and then we sequence the bit in between. Um, in terms of actually performing QC and assembly and typing, we're using the NF-Core viral recon pipeline with a few slight modifications to it, mostly just to make it work on our end. And since it's a general kind of viral assembly pipeline, some specific RSV um, uh, changes, nothing too major. And what we're looking to get out of that are subgroups, whether it's A or B, genotypes, which historically have been the genotype of the G protein, the glycoprotein gene. And then really what we're most interested in is doing, being able to call whole genome lineages and look at specific mutations. Everything that we sequence gets put into phylogenetic trees. Dan does a lot of that work with NextStrain's Augur pipeline. Sorry, I need to move. See. Okay, so the primer scheme that we're working with is the Danny Maloney scheme that came out a few years ago. We haven't really changed it. Each year we're kind of waiting to hear whether or to see whether we're going to have significant primer dropout. Other than a couple of primers that we've had to spike in additional material for, we haven't yet seen that. So it's still working well for us, even though it's now a couple of years old. It came out in December 2022. We were testing it a little before then. Um, and there's some papers that have come out of it. I'm not advancing. There we go. And then we're defining our lineages based on the big RSV paper by Stephanie Goya et al. From last year, or I guess 2 years ago now. And using this scheme that is implemented on NextStrain and other publicly available tools. So that's how we're calling our lineages. Again, this is the last 2 seasons. So the 2023-2024 season, and then the 2024 through the end of the respiratory season, 2025. We sequenced over 3,000 genomes, and they were almost a 50-50 split between RSV A and B. We did notice some patterns in terms of kind of the average genetic distance, the pairwise distance between our samples. So we saw differences between A and B. I'll get into that in a little bit. At the time, we had insufficient data to actually begin associating specific lineages with the severity of disease. Again, some of that is limited to the actual metadata that we have per sample. We don't always know. We can make some kind of coarse assumptions that, comparing hospitalized samples to not hospitalized, assuming that the hospitalized are more severe. But we're looking to add this year's data and then get some more metadata that's a little bit more granular in order to begin, doing some tests associating specific lineages or specific mutations. And then just kind of a fun tidbit. 16 or more of our samples were suspected co-infections. I say suspected because technically we can't rule out that there could have been some contamination after um, these samples were collected, where some RSVA got mixed in with RSVA-B either during extraction or amplification or sequencing. But we were seeing it enough, and this roughly matches a kind of 1 in 200 rate that has been previously reported in the literature, where individual patients were infected by both RSVA and B. And it's just if you work on the bioinformatics side of things, it's worth paying attention to because um, these can be you need some sort of practice in order to spot them before you assemble, or certainly before you take those assemblies and upload them to databases, because the assembly process will be agnostic to whether it was a co-infection. And it's very easy to assemble kind of a Franken genome of A and B samples. And yeah, I just kind of add that as a warning because I'll get into some of our comparisons to the global picture, and a lot of the QC we were doing was like, well, not a lot of it, but every now and then we were comparing our data to global genomes. We spotted global genomes that clearly were assemblies mixing A and B genomes, and were kind of poor quality and had to be weaned out. So we've got some flags. Basically, if we assemble a certain percentage of the genome that with our A reference and our B reference, we get a flag that if it's over, I think it's like 60% returned when we assemble with each of those references, that that could be a suspected co-infection. And we see these again at about a rate of 1 every 200 samples. We're also setting them aside. I have some thoughts about once we get a few more of these, kind of investigating those reads a little bit more closely, and seeing if we can take some insight out of those co-infections. So a lot of what we do, oh, and I should have said this at the beginning. If anybody has any questions, feel free to come off mute to raise your hand, and I'd be happy to clarify anything that's unclear. Definitely would like this to be a conversation rather than just me talking at you. But yeah, yeah, Curtis, there's one already.
Yeah, just to further ask about the A and B assembly portion,
Mhm.
like do you typically, run viral recon twice on a given sample with each A and B reference genome?
Yeah, that's exactly what we do.
Okay, cool.
We are agnostic. We don't have any knowledge of what they are before we go into the assembly step. I mean, we can infer based on kind of the number of reads that we get uh, after amplification with the A primers versus the B primers. But yeah, we still run everything through twice. We've
Okay.
tried some tools and had some ideas about splitting out reads. Uh, you know, by, say like running Kraken with,
Hmm.
A versus B. The problem is so many of the reads, or so much of the genome is identical or close enough to identical that we haven't had much success with that approach. So just looking
Gotcha.
at it after the fact has worked best for us. But I'm definitely open to other thoughts there. Um,
Yeah,
yeah,
I mean that would have been my first go-to as well,
yeah,
but yeah, they're close enough that makes it difficult.
it does. Yeah.
But yeah, I mean, and like when you run viral recon twice, is it quite obvious when you have a truly an A versus a B? Like, do you see those giant gaps in the GNF regions?
Yeah, and
Okay.
typically it's even more because we're both amplifying with A and B specific genomes, so we should
Hmm.
already be largely A if it's really A
Okay.
versus a small amount of, amplified regions with an A genome with B primers, for example. And then once you put it through another round of assembly,
Yeah.
typically, in terms of, and then, accounting for our coverage thresholds, which we're going for, 40x coverage, because I didn't mention this, but we're sequencing it all on ONT, so we tend to be pretty strict about the amount of coverage we're looking for to call a site. Um, and with all of that, it typically results in, a good quality. We don't. pass any genome unless it's over 90% in terms of the breadth of coverage.
Hmm.
And then, the opposite, the other one will be under 20% assembled. So
Okay.
it
Yeah.
tends to be pretty clear.
It jumps out at you.
Yeah, exactly. And
Good.
so that's why what I said before, when we get more than 60% of each, that's when we flag it as, this is potentially it could be contamination or it could be a co-infection. And we've, from the same sample, generated greater than 90% breadth from the exact same sample for A and B. Um, and those genomes are not perfect. Like I said, they sometimes are a mismatch of,
Yeah.
but they'll recover 90%. And so we separate those out and don't submit them. But yeah, it does happen.
Gotcha. Thank you so much.
Yeah, you bet. Um, okay, where was I? Yeah, so again, what we're doing is we're looking at, these time-scaled trees, and you can see, our sequencing efforts began in kind of, I think we might have some genomes from 2022 late in the season. We definitely have like spring of 2023 is when we really started kicking this off. So these genomes flagged in yellow at kind of the earlier branches in the tree. Then we had our first big season that's all marked in green, the 23-24. We tend to not get a lot of volume. It's a seasonal virus over the summer, so you kind of see a gap here. And then we again kick off. Obviously, the virus is out there and mutating and changing, but we don't see a lot of submitting volume here. So we see a little gap, and then we jump into 24, 25 with all of the samples. And um, yeah, we could have an updated tree with our 24 or 25, 26 samples. But again, that's an incomplete set. So I'm just showing you what we had as of June of last year. Um, Couple of patterns to take away from these kind of really high level trees are one, we're seeing an emergence of seasonal clades. So you can see this big clade here, almost half of what we were getting. This is RSVB on the right, by the way, and RSVA on the left. Almost half of the samples we sequenced of all of last year fell into this clade that we really didn't see. We saw some, a handful of related samples the year before. But this kind of came out of the blue with a bunch of new mutations that we were not seeing in our previous data sets and was the dominant clade for that year. There's a few more of these, like this one over here in RSVA. You can see my cursor, right? Um,
Yeah, we can.
cool. And then we also are seeing some things that indicate that we might have had a loss of a clade over the summer bottleneck. So, these clades were very common in 23, 24, and we didn't see any descendant lineages from them once we got to 24, 25. So, again, we're in the early, just 2.5 years of tracking this now, so it's hard to say exactly what this means, but we're keeping an eye on it and have some thoughts about, once we get a little bit more data, how we can investigate it. Um, we also, this is all Dan's work, but we have the ability to link some epidemiological data to RSV genomes. So this is a busy chart, but um, broadly, if you look at these final two bars here, well, I'll just walk through. Green bar, you don't really need to pay attention to. It's when we collected it. Um, the lineages are marked in the vertical bars in the second column. And then we have cases that are documented in RSVNet. So these tend to be more severe infections, like I mentioned with our reporting rules before, versus cases that were submitted to us, but were not documented, were not reportable necessarily. And then we had a small number of samples in this first year where we had documented use of nirsevimab as a treatment. So just a couple there. We're looking to expand this list. Um, and then over here on the right, there's a known mutation that I'll talk about in just a second, where we spotted that mutation emerge in um, in a few of our genomes here. So just talking about the nirsevimab briefly, this is a monoclonal antibody that tends to be administered for short-term immune boost. And there are some known from previous literature mutations in the RSV genome that can confer resistance to this antibody. And so we found a case where there were two hospitalizations that occurred after administration of nirsevimab. And this clade, identified here in the red bars, just zooming in on what we saw a few slides ago, shared this resistant mutation on the F protein, this K68N mutation. So again, it's really too small of a sample size to say anything other than, oh, we'll keep an eye on that. It's far from conclusive. But it's the type of data that we're starting to see emerge from, now over 2 years of sequencing, where we're moving toward this potentially being useful. Certainly, is worth keeping an eye on for any expansion of this mutation. Another project that we're working on, kind of along the same lines, is comparing the genomes that we're generating to global genomes. So for part of this project, like I mentioned before, we were pulling global data sets from NCBI and GISAID and kind of compiling these large data sets that we could use to compare to ours. So what you see here is RSV-A on the left and RSV-B on the right. The kind of a coarse scale lineage classification in the central colors on each of these trees, and then where we actually collected the samples. So anything on the outer ring that's in the darkest color was collected in Minnesota. Anything in a lighter gray is North American, but not Minnesota. And then anything in the lightest color is global, not North American. And you can see we have some clades that really emerged in Minnesota and kind of when put into the global context, look to be, Minnesota-specific clades, regional. I don't want to say outbreaks, but kind of had that appearance. Reason I'm hesitant to say outbreaks is we're just doing a lot of sequencing. So something that might look like an outbreak might just be kind of our normal landscape here in Minnesota. Not necessarily an outbreak, but certainly a regional clade that might be worth keeping an eye on. And it was kind of interesting. We saw a little bit more of that regional effect in the RSV-A genomes. If you compare this to the RSV-B, you would see that there were a handful, like this dark spot here, where we had, a clade that looked to be quite Minnesota specific, but broadly, Minnesota kind of reflected the global landscape rather than shaped it, if that makes sense. Through that. I will just say, we were a significant chunk of this data set. So from August 1st, 2023 is when we kind of began this comparative study through June 2025. Like I mentioned, we sequenced, over 3,000 full-length genomes. And when we compared that after some QC, that was, almost 20% of all the sequences globally that we could pull, that we could find anyway. So a pretty big fraction of that. That's percentage has declined every year despite us sequencing more and more, which is great because it means more and more people are also sequencing RSV and contributing to this project. Yeah, not just saying that to brag. I'm saying, you know, it's been fun to see this emerge as a more commonly sequenced virus, given what we're interested in doing with it. So if we just look at our genomes from the past 2 years, we can compare them along this chart. You can see we're representing a number of lineages, and many of them, emerge rather quickly and kind of dominate for that year, like, these short branch lengths out on the top right here. Dan was able to put together a lot of metadata about monoclonal antibody resistance. And so if we compare this outer circle, these dark bars on the edge here, these are genomes that are potentially associated with antibody resistance. So things that we're keeping an eye on moving forward. I don't have conclusions to share quite yet, but and the same thing for RSVB, roughly 1,500 genomes across now 3 years. You can see, like I mentioned, we've been upping our sequencing effort. This is just for the past 2 years. In the second year, we sequenced a lot more. Uh, and again, across both years, it was roughly a 50-50 split between A and B, with, slightly different dynamics. You can kind of stare at these and make of them what you will, but sometimes we saw a big A emergence early in the respiratory season. The next year, we saw B kind of take off a little bit faster. We're watching those dynamics, but don't have any kind of takeaways quite yet. Um, Again, we can just make some pretty charts with them and see which lineages are specifically, emerging or growing or declining throughout the year. These are these same charts, but broken down by lineage. Same thing for B. Again, I mentioned this before, but we saw a big emergence of a previously, unseen lineage in the second year of our sequencing. That's that B.D.E 1.5. I'll show you that one in just a little bit. So there it is at the top. And this really kind of highlights how the sequencing we're doing is shaping our global picture. These are all global genomes on Nextstrain. And this B.D.E 1.5 is really a Minnesota-specific lineage that has a lot of samples local to our, I don't want to say state, it's probably broader in the region, but because we're doing a lot of the sequencing, we're seeing it. If you've heard the old cliche about, searching for your keys under the spotlight, it's a bit of that effect going on. And the same thing with, a couple of other clades like this 1.6 tends to be concentrated in Minnesota. Um, we also kind of see a shift in the broadest or kind of broad pairwise comparisons between our genomes each year. They tend to, this is kind of just taking these trees and representing them on a 2D axis of pairwise differences between every individual sample. And you can see A and B do have some different dynamics. It's, I think, reflected in that earlier tree that I showed you where A tends to be a little bit more uh, branched out. There's genomes that tend to be more different from other genomes broadly. We're seeing higher levels of differences, pairwise differences between those genomes and the rest of the genomes that we're sampling, compared to B, which tends to be kind of more reflective of the global picture and more similar. Each individual genome is more similar to the other genomes in the data set. I'm going to skip past this. This is the same data, just kind of presented in a different way. We did see some differences in terms of the seasons, though. And the B seasons tended to resemble each other a little bit more, and A had a little bit more variance between season. Um, like I mentioned before, these are these same plots, but again, as we compare 2 years of data, we're observing these seasonal losses in clades across the tree. We're seeing emergence of different clades across the tree. Same thing for B losses here and emergences specifically. This one that I mentioned, B.D.E.1.5, really stood out in our second year of sequencing. And so that led us to ask a question. Sorry, I'm just. Oh. Um, uh, Yeah, so that led us to ask some questions about, how could natural selection be shaping our RSV genomes across each year? So we've noticed seasonal changes in the clades and lineages that tend to precipitate. We have a lot of data. And so, we can ask this question, can we find evidence of site-specific positive selection in RSV genes? And so we started to so again, as I mentioned, we pulled complete genomes that were, subject to roughly the same QC that we uh, routinely do on our data from September 23 through May 24, and then September 24 through May 25 from GISAID. We filtered anything out that had, again, more than 10% missing data, the same standard we apply to our routine data. And then we performed alignments. We did a little bit of manual inspection, weeding out, for example, contamination or co-infections. And then we put together 5 unique data sets for each of A and B, so 10 total data sets. And they broadly took this pattern. So we took a complete data set with all of the genomes that we pulled. We took that same data set and removed Minnesota genomes. We took a data set with the just North American samples. We took the same data set and removed Minnesota samples, and then we have just our Minnesota samples. And I'll show you why we were interested in doing that. Because these data sets let us ask different questions. So when we look at selection, we can say, if we take the whole data set, how are global genomes being shaped by selection? We can also say, how are North American or just Minnesotan samples shaped by selection? These are high-level questions. But we can also compare these data sets, and we can say, are there sites under selection in North America, but not the rest of the world? Things that are emerging here that might be, potentially subject to regional selective pressures or new mutations that are emerging in North America and, being positively selected throughout our for whatever phenotype, but that have not yet spread to the rest of the world. Same thing, comparing North America to Minnesota. We can also compare across and say, how do Minnesota genomes affect the global picture? So if we introduce our Minnesota genomes to the global landscape, are we then seeing things that either do we now see sites that are under selection that we did not without the inclusion of Minnesota genomes, or vice versa? Did Minnesota dilute the global picture with all the genomes we're putting into it to the point where we're actually masking sites that could be under selection, reducing the kind of how much they stand out. Same thing here, same question, but just on a more local scale. How does Minnesota's inclusion affect kind of the picture of selection in North America? So we can do these comparisons year on year and by RSV A and B. We can also ask, are any sites that are under selection in one year changing the next year? Are they, part of these, did we see a site that was under selection that's associated with a lineage that really took off the next year? Or was something under selection and then lost in the following year? Asking these questions. So you can see it's a ripe data set to come up with questions. Answering them is the harder part. So just to kind of take a first pass at this is, not an exhaustive approach, but we thought we could do a gene by gene or really a site by site analysis across each data set, look for positive selection. We just made a first pass looking for any site that showed a base factor greater than 15 after we applied this kind of fast approach for inferring positive selection, called FUBAR. I refer to it again. And so if we just look at, I'm just going to narrow in on kind of the key suspects, this G protein, the glycoprotein function, because it tends to be, again, mutagenic and has a lot of, past evidence that it's, the gene under selection if we had to pick one. Obviously, there are sites under selection on other genes, but just narrowing in. In our global data set, we found 24 sites that passed our threshold in 23, 24, just within the G gene, 14 of them in 24 and 25, and 10 of these were shared across seasons. So they tended to, we're kind of keeping tabs on those. As you'd expect, a few fewer when we compare our North American data set, but again, 8 of these that were shared across seasons. And then there were within our Minnesota specific data set, we did find, 4 of these sites and 6 of them under selection in Minnesota across each season. Again, just throwing a lot of data at you for now, but if we subset Minnesota out of this picture, you can see we lose a few of these. So we went from 24 down to 21. So Minnesota's inclusion, added 3 sites to the picture, to the global picture, or added, 3 sites in 23, 24 to the North American picture, or 5 in the next year. So you can start to see how, our sequencing efforts add to the our inferences that we can make. And finally, this is ongoing work, so I I wish I had a little bit more to report to you now. We're doing some lit reviews of the sites under selection, but you'll have to look out for that and, a forthcoming publication or talk. But we do now have a short list of 50 sites that I think are pretty interesting because they are either unique to Minnesota, as in, we're seeing selection um, only in our Minnesota genomes, but not in our other data sets, or they seem to be sufficient or I should say, these partially overlap, sufficient to drive selection by inclusion in a broader data set. So these are sites that, we see under selection in this data set, but we do not see in this data set. So it's the contribution of these Minnesota genomes to this broader picture that is kind of uncovering them or allowing us to infer the presence of positive natural selection when we include our genomes. So now we're taking these sites and we're looking through the literature to try to find any sort of, potential mechanism for why they might be under selection. And in a few cases, we have come up with, I would say maybe a third of these 50 sites, we've found some evidence of previous reporting of that site being found to be under selection or associated with antibody resistance, etc. And this is across all of the genes, not just the G protein. That's why the number differs a little bit from what you saw before. So just kind of wrapping up on where we're at this point. We do think that, the work we've been doing is shifting the global landscape in the study of HRSV. So we're discovering new clades that, we only detected through our sequencing efforts. We're pairing this with the RSV net data and classifying each infection as severe versus more moderate or mild, and starting to use that data to look at, any sites that might be associated with severity of disease. And then we're trying to get additional data of genetic studies of RSV mutations that, are emerging during or between infections. And I think, there's some element of this that's influencing strategies with both internally and externally of other viral pathogens. And so, again, we're seeing evidence of positive selection. There's more of this to come as we continue to work on it. But we do think right now we're adding to the global picture, and we're always looking to expand our sampling. So I'm not sure who's on this call, but if you're, a potential submitter, we'd love to have genomes. We're always looking for more. Of course, with more data, we want to, continue to think critically. We, again, are trying to ask this question of, can we identify potential outbreaks before they happen or as they're happening so that we can intervene? That's tricky given our, submitting practices right now, but we're not saying it's impossible. Again, can we leverage any of these mutations that might be linked to severe infection? We have some ideas about, GWASs in the future, that sort of thing, to look at, mutations associated with severity. And then can we track any emergence of mutations that are linked to antibody resistance or vaccine escape? You saw, we've done this a little bit, but the sample sizes were pretty small, so we're looking to expand that. And then this is really kind of getting to be more and more of an outdated question. These slides are kind of an evolving thing, but at one point we were trying to push other states to expand surveillance beyond COVID, and now a lot more states are. So I think next time I give this talk or a version of it, I might have to edit that question. But if you are in a state that is considering sequencing, we think you should, should sequence RSV. It's a fun pathogen to work with. So again, here's our team. If you have any questions, feel free to ask now. I've got a few minutes. Or if you're interested really on the genomic epi side, Dan's a great person to talk to about this. I will pause for questions, but I did just want to use this platform real quick to just say we're in Minnesota and it's not great right now. If you've been following the news at all, we're seeing, masked federal agents knocking on doors, barging into houses, arresting, I can't even really say arresting, abducting kids as young as 5 or 2. And today is uh, I'm working until I log off this meeting and then I'm out for the day to go march. It's really damn cold here too. It's currently -13, and there's going to be thousands of people out on the streets fighting for this. I don't think science is or ever has been apolitical, so I wanted to use this platform to just say if you're observing this at all, to think about what might happen if this happens to your community, and what you will do, and if you yeah, have a chance to use your platform to do something positive, I would encourage you to do so. And if you can't use your platform to use your finances or your time in some productive way, because I think this is really important right now. So that's all I'll say. I'll jump back to the question slide and then wrap up. Try it. There we go. Well, thanks everybody.
Yeah, I don't have a question, just a comment or two. Just awesome work. I really appreciate you sharing all this. This is a pathogen that at least that I've seen that hasn't been as deeply studied, especially in the public health context. So this is really awesome work, and the fact that you're sharing all of this data is incredibly valuable. I mean, I remember a couple years ago when this project first started up, we were looking to test out RSV data with our SARS-CoV-2 pipeline, and immediately saw that your group had posted the reads test array, and I was like, perfect. I know I can trust this data. Like, let's test our pipelines using it. So that's just one application of this data, and I just applaud you guys for your efforts and doing all this work and sharing it. So yeah, thank you.
Yeah, thanks. Thanks. It's glad to hear. I'm happy to hear that it was useful to you. We will continue to generate it and submit it as long as I have to say anyway, as long as we're funded to do so. So more to come. And if there's no other questions now, I'm happy to give everybody 15 minutes back. Appreciate you all hopping on. Uh,
Yeah, thank you so much for that. That was really interesting.
Stop
Have a
my
good day,
share.
everybody.